Hi, welcome to the Radiation Research Society Vodcast. I'm Allison Burell. I work at the National Science Foundation, but I help out here with the interviews. So I'd like to welcome you guys to a roundtable discussion. Would you like to introduce yourselves?
Sure. I'm Charles Limoli. I'm a professor of radiation oncology at the University of California, Irvine, and am very interested in doing a lot of the basic science work that is involved with the flash radiotherapy.
So hi, I'm Marie-Catherine Vozenin. I'm the head of the Radiation Oncology Laboratory in Lausanne at the CHUV, and I've been doing pioneer work about the flash irradiation.
Great, and so today we're going to have a roundtable discussion on this new up-and-coming technology, flash radiation. For our audience, could you explain to us what is flash radiation? This sounds so interesting.
Well, it's a new approach in radiation oncology that enable us to deliver a dedicated dose in a very short period of time. And when I say very short, I mean less than 200 milliseconds, whereas usually we use minutes of irradiation.
So it's been particularly useful in uncovering some, well, I must say, rather unexpected effects. And the unexpected effects come with reducing a lot of the normal tissue toxicity that comes with the more conventional dose rate paradigms they use in the clinic. And this has caught the field by surprise. And everyone's kind of scrambling to figure out what's going on. And largely due to the work of Marie-Catherine, it's really caught the imagination of the field.
How did you guys come up with the idea to do this less than 200, what did you say, how much was the time again?
200 milliseconds.
Less than 200 milliseconds.
Right.
To be clear, this was her idea.
Yes,
so,
I know.
Yes, okay.
Yeah, so this is amazing, but so...
So you mean how
I'm just
the idea
curious
was coming?
Yeah, where did this idea...
In fact, it was pretty simple. We do have access to an experimental irradiator that was built to study radiolysis. We never used that before for any experimental purpose or biological purpose. But we thought it would be interesting to have a look at that, and that perhaps this very short term of irradiation would induce a differential response in the tissues. So we started irradiating experimental animals and came up with very surprising results. As Charlie said, it was really unexpected. At first, we thought we do have a problem. We have not irradiated those animals. It's just not possible. We don't have any toxicity, any of the toxicities that were used to get after irradiation.
So when you started with this, you were just, there was no cancer involved. It was just, let's irradiate them with this technique and see the toxicity and look at the toxicity. And so the amount of radiation that you use, though, is much higher than normal therapeutic doses, correct?
No, it's much lower.
Oh.
But we are usually, well, we are normal tissue people.
Okay.
The two of us are working in the normal tissue field. So what we're currently, well, basically using is single dose of irradiation. That's the main difference with the therapeutic irradiation. Okay. But it's single dose, large dose, but in total is less amount of irradiation than a cancer treatment.
Okay, I see what you're saying. So the radiation exposure that's received by the animal is a lot lower.
No. So there's two things.
Sorry.
When you treat people with radiation, you target a tumor and you prescribe a dose, the total dose, that can be anywhere from 60 or 70 gray to as little as 1. And the rate at which that radiation is delivered usually is about what we call 2 gray a minute. Okay. And so now she's been using a technique that delivers the dose rate, not the dose. The dose rate is ultra fast. Mm-hmm. And so when I first heard her results back in 2014, I was skeptical, as many people in the field were. And so I said, hey, I did a lot of stuff on normal tissue injury in the brain. So we sent her some animals, and they came back. They were given 10 gray, which causes significant neurocognitive complications. Right. Normally, when you give it at the regular standard rate. Dose rate. Mm-hmm. Nothing happened to these animals at 1 month, 2 months, 3 months. I go, come on. 6 months, no neurocognitive complications.
Just from that first initial dose.
Wow. And the animals that were given that one dose on the standard dose rate showed all the usual things we've been trying to fix. They showed all these types of deficits in learning and memory, mood disorders involving depression and anxiety. Lo and behold, much to my amazement, 6 months later, the animals were given this ultra high dose rate. None of these neurocognitive complications. So she's done a lot of stuff before in the lung and the gut and all that stuff. The field of radiobiology has been trying to fix neurocognitive complications for 4 decades with dismal success. And to actually find something that you can fix that happens in less than a millisecond is amazing. And so there's your functional impact in the brain. Mm-hmm. And in the other normal tissues, endpoints that have been equally recalcitrant to being fixed, like fibrosis, they don't happen. And so, and then, of course, you asked about a tumor, and then she has the story with tumor, because now, if I'm telling you all these great things about all the normal tissue sparing, all right, so then you're going to spare the tumor, so what good is it? But
it's not the case. In fact, when we do irradiate the tumor, we do have isoefficacy. So it's really specific to normal tissue. It's not specific to any organ. It can be seen in the lung, in the gut, in the brain. It's not specific to specific spaces. Mm-hmm. It can be seen in mice, different strains. It can be seen in pigs. It can be seen in domestic cats. We've been irradiated domestic cats with squamous cell carcinoma. It can be seen in fish, zebrafish. Wow. So it's a trans-species observation, but specific on the normal tissue. In fact, specific on tissues that are normoxic.
So you mean specific as it's not affecting those tissues?
It is beneficial. So the key points here are that not just our groups, mm-hmm. but other groups have also found these beneficial effects. And there was a lot of concern that the dosimetry was maybe off a little bit, and that's certainly not the case. So the effects that we're measuring are delivered at isodose with differing dose rates, and now every model system we've touched on, even cell-free systems where we look at the production of hydrogen peroxide, each system we look at, going from no cells to zebrafish to rodents to pigs, cats, all show this. Multiple groups show this benefit. And to the best of my knowledge, I don't know, this is one of the first times I've seen something that stands to revolutionize the field of clinical radiation oncology, where you have your most robust defined effect in vivo. And now we're trying to deconstruct the in vivo situation to figure out what's going on. Mm-hmm. Most of the time in this, you make your discoveries in yeast, worm, and flies, and move up to cells, and then, oh, it didn't work in animals. And this time, wait a minute, we have all of our functional effects in animals, so it's like, it really doesn't matter why it happens, but it does. I mean, we have something that works. And figuring out exactly what the basis of this is going to be fun and challenging, yes. But it's usually the other way around, right? Well, you killed the cell, what about the animal? And now we have the big effect in the animal, and I think you're actually almost poised to do something with humans now, right?
Right, and that was my next question was, is this going to move into clinical trials? And I had heard that one of the barriers to that was the mechanism, was that
Well, I don't think that the mechanism is a real problem because we are using radiation therapy and ionizing radiation since more than 100 years, and we really don't know everything about the mechanism of ionizing
That's what
radiation.
I was thinking, that there's lots of drugs out there that we don't fully know the mechanism, but we know that it works and it passes through clinical trials.
I think the main critical point for the clinical transfer is to be sure about the safety. Mm-hmm. And is to be sure about the right selection of the patients that would benefit from it. So that's the thing. The very good point for us in Lausanne is that we have an incredible team of physicists, mm-hmm. and an institute that is dedicated to all the dosimetry and the metrology, and we have in the team metrologists that are doing the dosimetry, so we are really accurate with the dosimetry. Claude Bella is the guy that is doing everything. Mm-hmm. So we know from safety point of view that our dose, the dose that we're going to prescribe, is going to be accurate. And from this point, we hope to be able to move forward to the clinical situation. It's going to be probably one of the first clinical trial in radiation oncology, because there is no trial per se. I mean, phase 1, phase 2, phase 3, it's always combination between irradiation and drugs, mm-hmm. but there was no trial for the proton, for instance.
Interesting.
So
I didn't know that.
It's a completely new concept.
Yeah, there's a lot of combination therapy, chemotherapy, radiation.
Right. And when you look at all the effort that the field, the radiation oncology field, spent on hypoxic cell sensitizers, modulating oxygen tension through a variety of techniques, these things have met with some success, but they've taken years and years to develop, and even these drugs, hypoxic cell sensitizers, they fail in some trials because the selection criteria was maybe a little bit wrong, and so now if you consider that all these chemical and biologic interventions, how long they take to develop, and now we have something that we can initiate and it's over and done with, instantaneously. Yeah. And we still get the long-term benefits without compromising tumor control. I mean, really? Yeah. I mean, this is really, and frankly, it's astounding.
So normally when a radiation dose is prescribed by a radiation oncologist, like you said, sometimes it's not all given at once. It's given over a period of weeks. In different doses, but this is something where you would just give it one time, and it would be maybe more than one time. Yeah.
Well, it will depend upon the tumor we are going to target. Mm-hmm. So far, all the experiments we've done with experimental system and domestic animals with cancer, we have applied only one fraction with success. So with this technology, we are able to apply very large dose that controls the tumor without toxicity. But in human, we are probably going to be able to make fractionation studies, and we already know that it has no impact on the efficacy of the flash radiation therapy.
So one way to think about it, right? So depending on where your tumor is, mm-hmm. every clinical oncologist will dose that tumor to the maximum tolerated normal tissue level. Otherwise, you're running the risk of not controlling the tumor, and it would be medically irresponsible. So now, and let's say a typical brain tumor treatment would involve 32 gray fractions. So the patient has to come in when a fraction's going to the doctor's office on a day. Mm-hmm. So now if we tell the doctor, let's suppose we can even cut that in half, mm-hmm. or a third, or a quarter, and we can increase the dose to the tumor and keep the normal tissue toxicity the same or better. There's nothing really at risk here as long as it can be delivered safely. So we've cut the cost of healthcare. The patients don't have to see the doctor, which I'm sure they love to do. And so you can theoretically dose escalate to every normal tissue bed with a lower number of fractions, a higher dose per fraction, and keep the normal tissue complications the same or better. I mean, there's not many things that you can do that give you that kind of an outcome.
I mean, that's pretty amazing. Is there an instance in radiation oncology where the maximum dose to a tumor might not necessarily be the maximum dose that would be needed to completely eradicate the tumor, but because of the toxicity, you can't go any further?
Well,
right?
if you dose a tumor too high, I mean, you can kill a patient if the normal tissue toxicity is too high. So, I mean, there is a point where they have to stop.
You have to stop, but the tumor keeps growing.
And that's where all the clinical knowledge comes into play. I mean, I'm not a clinician, but I mean, you know, we don't cure 100% of tumors with radiation therapy, but it's very useful.
Yeah.
So.
However, to be clear, we still have technological limitation.
Okay.
So it's going to take a bit of engineer work to be able to have devices that's going to be able to treat any kind of tumor overall the body. Currently, what we have is devices that can treat superficial tumors, but probably not the internal one.
Okay.
Our colleagues in Stanford are currently working on the question, and they do have pretty nice ideas how to solve the technological problem.
That was what I was wondering. I'm glad you mentioned it. Was the technological issue? Would it be? Is it the same types of machines that are used now to deliver radiation? Would they need to be modified, or?
I mean, there's nothing that's fundamentally different, but the components of the machines do need some upgrades, and you know, basically we need to convert the current clinical landscape into machines that deliver a high enough energy so we can penetrate to the depth desired and provide the dose rate necessary for our normal tissue sparing effect. Right now, we have the energies that can penetrate, but they can't deliver the fast dose rate. And her machine in Switzerland has a beam that delivers the dose rate, but lacks the penetration depth. And so, but you know, we're going to try to send someone to Mars in 20 years.
Oh, yes,
we can get this done in well under 20 years.
I think that this technological problem is not.
No.
Correct me if I'm wrong, that there's nothing that really hasn't been invented yet. It's just kind of putting all the right pieces in a different configuration. So there's not a magical piece of technology we're waiting for someone to invent. It's just a matter of money and getting it done. So
Reconfiguring it. Okay. So my next question is to tumor types. So when you started working with these model organisms like the pig or the cat, what kind of tumors were you using? Were they and were they ever xenografts? Did you ever work with xenografts?
Well, we've been using a lot of solid tumors, all kind. Xenograft, yes. Orthotopic tumors, spontaneous tumor in transgenic animals, and also spontaneous tumor in the domestic cats. So yeah, all kind of solid tumors.
Oh yeah, I think a colleague of mine had mentioned something with a tumor on the nose. Would you want to tell that story?
Yeah, squamous cell carcinoma. So it's a typical disease in white cats, and they do have the development of this squamous cell carcinoma on ears and at the surface of the nose. For us, it was a very good model because it's superficial. It was also very interesting because the outcome is pretty poor. The animal can get surgery, but then once they don't have any nose anymore, they are not smelling. If they are not smelling, they are not eating. If they are not eating, they just basically die. So here the question was to really try to improve the treatment for those animals. Conventional radiation therapy can be used and is used, but it's fractionated, so it's 20 fraction. Usually those cats are pretty old, like 10, 12 years old. So 10 anesthesia in a row for 2 weeks is pretty difficult to handle. Here we were just proposing very, very simple, one shot. irradiation, one-shot anesthesia, less than 30 minutes of anesthesia. So it was pretty easy to convince the owners to just try it, and the outcome was just perfect. We had no toxicity. The nose was fully reconstructed after the irradiation. The only side effect was depilation, which is not a major issue as compared with.
So they lose some of the hair?
Yeah,
around
exactly
that
at the
area?
field of the irradiation.
Okay.
And this is published. It has been published this
So
summer.
it's almost too good to be true.
It does. It sounds like this.
It's an exciting time
Very,
to be
very
doing
exciting.
the basic science and listening to clinicians grumble about, well, you know, but I do think that we're going to be able to do some good for a lot of people in a few years.
I'm really excited to see about this. So yeah, primarily, I mean, it makes sense that this would be a treatment for solid tumors. Is there, I'm not familiar, because I'm not a clinician myself, but is there radiation therapy that's used on non-solid tumors?
Right.
There is. So, but something like this is more like a targeted therapy, but do you see it being used at all in non-solid tumors?
Oh yeah, well, yeah.
Why not? I mean, we irradiate the whole brain of rodents, but I mean, I think what you're getting is systemic disease is certainly more difficult to treat with radiation.
Like blood cancers.
Well, I mean, right, in any stage 3 or 4 cancer is more difficult to treat
Yes,
with
or
radiation alone. It's usually
metastatic,
combined with chemotherapy. But no, there's nothing that limits it.
Glioblastoma that's kind of spreading out throughout the brain.
Well, I mean, they still give whole brain for that too. And if you can deliver the whole brain with flash, then it's going to be, it certainly won't be worse than what we're doing now.
Right. So that's a possibility, doing I mean, not necessarily whole body irradiation, but.
Well, I think the term you would use is we can treat larger volumes.
Larger
Right.
volumes.
Which is
Yeah,
really
'cause-
something that limits the more conventional treatment. Right, if you treat your whole liver with a certain dose of radiation, I mean, you need some of your liver to work.
Yes.
There's you can get rid of 30% of it, maybe 50, but you can't get rid of 70 and 80% of it. So, I mean, if we're able to treat a larger volume and minimize that toxicity, then that makes all these treatments more feasible.
The idea then is also to be able to merge this short time of delivery with all the new imaging approach. So if we are able really to merge those two things together, we're going to be more efficient, that's for sure. And we can be very precise also, even in blood cancer like Hodgkin lymphoma, for instance.
So what are your next steps? You say you would like to move into human clinical trials. Do you have a specific type of tumor that you want to start with?
Well, yeah. For now, we're gonna target the superficial tumors, so mainly cancer tumor, skin tumors.
Yes.
Sorry.
That's okay.
But
Like
then,
squamous cell.
like some type of basal or some types of cutaneous lymphoma, for instance. Then the next step's going to be for us in Europe to investigate intraoperative radiation therapy. In that case, it's going to be single-shot irradiation, and the target's going to be head and neck cancer because they are infiltrating with large vessel infills. So if we can protect the large vessels, we're going to be able to provide a large field of irradiation. It's going to be the same for pancreatic cancer. So those are probably going to be the two next major tumors we're going to target. The third one is glioblastoma.
Yes.
Of course. That can also be reached with intraoperative radiation therapy. And to prove that it's going to be feasible, we will have a phase 2 clinical trial running in dogs with Swiss vet schools of Zurich and Bern. So we're going to try that, start that in the next few weeks. So
you're based in Switzerland?
Yeah.
So these trials are going to start in Switzerland?
Well, I guess yes, because we are the only one to have a machine running, up
Okay.
and running right now.
And in the background, we have a lot of preclinical models that we're testing, mechanism, the optimal dose per pulse, and things like that. So we're going to be optimizing the actual paradigm using mice, rats, dogs, and getting it, increasing our basic understanding of the mechanism and the physical chemical parameters that are important, so that when we hit these human trials, we're armed with all the knowledge we can get.
Right.
But as I said, the US won't be far behind, because there is a lot of technologies that is currently being optimized. So,
Like with your collaborators
right,
in Stanford.
Right. So I think that it's going to be
Yeah.
kind of spread out very quickly.
Yeah, I would hope so. I mean, ideally, it would be nice to have all of these trials going in tandem.
Well, well, this is exactly what we want.
Yeah.
I mean, I'm sending the animals and things to her in Switzerland, and we're doing a lot of stuff back at our institute. But I mean, really, the more places that this gets launched at, the quicker it's going to get implemented, and the quicker people are going to believe this. I mean, this is no good to have it one spot.
No, sure.
No, the idea here is to share, right? We
Yeah,
want
yeah.
to improve the clinical outcome of people with cancer, and so I think that once we're close to having a lot of nice publications come out, and people she hosted a nice workshop in Switzerland, and the NIH hosted a nice workshop in Washington. Mm-hmm. So it's getting some attention, which is really the first steps in all of this. And with the papers and all the workshops, and with more people thinking about it, I think really it's safe to say it's a matter of time. And the quicker we can make it happen, it would be the benefit to all. So
the other very interesting aspect is that we are all academics.
Right.
So we are used to work together, share the knowledge, and try to push forward. So I think that this is the main, perhaps the main strength of this new approach. The fact that we are not dealing with the commercial issues.
And I think that's what I love about science, and why I'm a part of it, is that it's international, but as scientists that we don't see borders. We see problems, and
we're
we
not
see
trying to make money off this.
Right,
Maybe
exactly.
earn a
We
living,
want
but we're really
to share
wanting to get the knowledge out there.
Exactly.
And on the side, it's quite interesting as well. I mean, why this works. So that's what makes it fun.
Yeah.
So.
Well, this is very exciting. Are there any other aspects of this project that you'd like to talk about?
I'm pretty good. I think we're doing what we can, and the right people are listening, and you've got a lot of nice stuff going on in Switzerland, and there's, as I said, there's places in the United States at Stanford that are well on their way of getting this going, and we have lots of good data ready to publish, and I just think really it's just keeping the momentum moving forward, and I guess if there's an obstacle outside of some technological obstacles, it's just keeping the funding coming in so that we can keep generating the data. But I mean, that's going to happen. So,
Right,
I mean,
right.
it's
There's always
there's always a grant to write. It
funding
doesn't matter what you
hurdles.
do. That's what we do, and they'll get funded eventually. And so I mean, but that's the scientific process.
Yeah, but this definitely sounds like a transformative project.
Well, it is, and all this stuff has to survive peer review, and there's people that won't believe it until the last minute. I mean, that's fine. I mean,
Yes, that's a game.
if everyone agrees, then you kind of look in the mirror and go, uh-oh. I mean, I'm not sure something must be wrong.
Something must be wrong. Yeah.
But so it's a good check and balance.
Yeah, just to say that this observation has already been done 40 years ago by other people, but then
But
was
it
not.
It got stalled.
Yes, it just gets stuck in the way. So,
so
we
disappointing.
just rediscovered that and went back to the old literature, so that. Some people here in the US, in New York at the Memorial, and in Europe at the UK were already thinking about such kind of approach. So it's just like we're not discovering anything new, we're just going.
Oh, you're being modest. But yes, I'm excited to see where this moves forward. And I know we have 2 radiation conferences next year, the International Conference for Radiation Research, and then
The one at Manchester, right?
Manchester. And
They'll be having something on this there, I'm sure.
yeah. So the Radiation Research Society Vodcast and podcast is going to be interviewing and having interviews at both conferences. So hopefully next August maybe we could interview you guys again and hear some.
We'll actually both be there.
Great.
So
Great.
I'll be
All
talking
right, I'll
on
put it
something
on the
very
schedule.
different, but it's still, I'll be very interested in, I'll be doing this for many years, so.
Yeah, I think we do have sort of 20 years ahead.
So anyway, so hopefully this kind of demystifies some of the flash and maybe demystifies or at least justifies some of the high expectations. But you know, it's been a while since I've seen something come along that has been, has this type of potential promise. So I'm excited as well. Makes it fun to being science. So.
And I have to say, I like the name FLASH. I'm happy that it's not an acronym because everything is acronized these days. But it also, you know, is kind of a double entendre. Like it's FLASH. It describes exactly what it is, but it also can describe how it's treating.
And if you want to put her movie of the animal being irradiated, that's exactly what you see, a flash.
Yeah.
I mean, it's exactly what it is. So anyway.
If you have a link to that, we could add that to the description for this podcast. Yeah. Well, thank you guys so much for coming and talking with us here at the Radiation Research Society Vodcast. And like I said, I look forward to hearing more from you in the future.
Very good.
Thanks
Thanks for
a lot.
having us.
You're welcome. Thank you.